Why has no human ever lived beyond 125 years? This question has fascinated scientists and the public alike, prompting a deeper look into the biological limits of human lifespan. Despite advances in medicine and improvements in living conditions, there appears to be a ceiling to how long we can live. In this section, we explore the factors that define these boundaries and what they reveal about the nature of aging itself. The Hayflick Limit refers to a groundbreaking discovery made by scientist Leonard Hayflick in the early 1960s. Through his research, Hayflick demonstrated that normal human cells are not capable of dividing indefinitely. Instead, they have a finite number of times they can replicate before growth slows and eventually stops. This finding challenged the prevailing belief that cells could divide endlessly under the right conditions. Hayflick's work laid the foundation for our understanding of cellular aging and has had a profound impact on the fields of biology and medicine. Cell division is the process by which a single cell splits to form two new cells, allowing organisms to grow, repair tissues, and maintain healthy function. In humans, this process primarily occurs through mitosis, where the genetic material is duplicated and evenly distributed between the daughter cells. Unlike certain organisms, such as some plants or simple animals that can divide indefinitely, human cells have a built-in limit to how many times they can replicate. This limit, known as the Hayflick Limit, is largely due to the gradual shortening of telomeres, which are protective caps at the ends of chromosomes. Once these telomeres become too short, the cell can no longer divide, ultimately leading to cellular aging and contributing to the finite lifespan of human tissues. Telomeres are specialized structures found at the ends of chromosomes, acting as protective caps that safeguard our genetic material during cell division. Each time a cell divides, these telomeres become slightly shorter, as the replication process cannot fully duplicate the very ends of DNA strands. Over time, as telomeres progressively shorten, they eventually reach a critical length where they can no longer protect the chromosome. This triggers cellular aging, also known as senescence, and ultimately leads to the cell's inability to divide further, resulting in its death. The gradual shortening of telomeres is a fundamental process linked to aging and the natural lifespan of cells. Human cells divide a finite number of times, a process governed by the shortening of telomeres, protective caps at the ends of chromosomes. With each cell division, telomeres become progressively shorter, eventually reaching a critical length that signals the cell to stop dividing or to die. This mechanism helps prevent uncontrolled cell growth, but also limits tissue renewal and repair as we age. Research indicates that this gradual telomere shortening is a key factor in setting the upper boundary of human lifespan. As a result, the maximum observed human lifespan of around 125 years appears to reflect the point at which cellular renewal can no longer sustain vital functions, establishing a biological ceiling for longevity. Current scientific research into extending cellular lifespan focuses on understanding the mechanisms that govern cell division and aging. The Hayflick Limit, which describes the finite number of times a normal human cell can divide, is a central concept in this field. Researchers are investigating the role of telomeres, the protective caps at the ends of chromosomes, which shorten with each cell division and contribute to cellular aging. Studies are also examining the enzyme telomerase, which can extend telomeres and potentially allow cells to divide beyond their natural limit. By analyzing these processes, scientists aim to uncover safe methods to maintain cellular health and delay aging, while carefully avoiding unproven or speculative approaches. In summary, aging is not just a random process, but one that is intricately programmed at the cellular level. By uncovering the biological mechanisms that drive aging, we gain valuable insights into how our bodies change over time. This deeper understanding encourages us to prioritize improving health span, the period of life spent in good health, rather than merely seeking to extend the number of years we live. Focusing on health span means aiming for a life that is not only longer, but also healthier and more fulfilling. As we consider the boundaries set by our biology, it's important to reflect on the quality of the lives we lead. Longevity alone does not guarantee fulfillment or happiness. Instead, embracing our limitations can inspire us to make the most of each moment. The true question is not how long we live, but how well we live within our biological limits.